course, approximately how much time to produce detectable antibody levels in the blood. So why? The answer is in 1 week because the range for the primary immune response is 2 days to 2 weeks. But specifically, it has something to do with the kinetics of the primary immune response, wherein the inductive or the intent or the lag phase is usually at the 5 to 7 days. That's why it's 1 week. So in the lag phase, what happens is the antigen is recognized as foreign and the cells begin to proliferate and differentiate in response to antigen. So that's why at this moment, that's why there is an increasing levels of antibodies up to such time that it will be detectable in the blood. So yeah. It's another thing is the cerebral version. So you know that it's the development of the detectable specific antibodies. So that's why you can see the detection there.
Okay, thank you. Agree, disagree?
Agree!
Did you receive the handout? Yung handout ko?
Question?
Immune 1 and Immune 2? Wala? Oh, congratulations. Okay, I give that to the two presidents.
I actually announced it na nasa photocopier sa labas. That's alright.
Okay, I assume they are absent. Okay, next question. Oh, I thought you transferred all the questions in one laptop. Like what Second 8 did, para tuloy-tuloy. Kasi pag lipat yun, 5 minutes na yan.
Alright, just to answer question number 15. Question number 15 is, during the maturation of B cells, B lymphocytes, the first immunoglobulin heavy chains, it is the black, and explained. After what we have read, we found out that the answer is the mu chain. Mu chain is distinct for immunoglobulin M. For example, why it is distinct? For example, IgG is determined by the distinct heavy chain called the gamma chain. Then the IgA is also distinct because of the IgA is has a distinct heavy chain called the alpha heavy chain. Now, the mu chain is distinct for IgM. It has a crucial function that the mu chain is the antigen receptor for the IgM. And then this serves as a binding site for the antigen to form the antigen antibody complex. And then when this complex is formed, there is this what we call a cascade of forming a lot of antibodies to fight for a specific antigen, for a specific infection. And in this side, you can see that, let's focus on the mature B cells. Its surface marker is the IgM. Then the function of the mucin is that it is the specific receptor for the IgM. So every immune response starts with the synthesis of the mu chain. Thank you.
Okay, agree? Disagree?
Puta mo tiyak. I-record mo daw siya. I-record kita. Puta. Si Hani mo ng kwan diri. Covered. Okay, record mo daw.
Thank you.
Our group is tasked about the
Post niyo ba?
question number 16, which is the delayed hypersensitive reaction is characterized by and explained. A delayed hypersensitivity reaction, first, what is the delayed hypersensitive reaction? The delayed sensitivity reaction is a major mechanism of defense against intracellular pathogens. Pathogens are which organisms that is harmful to our body. Example of which is your Mycobacterium tuberculosis, which is responsible for your TB, and fungi and parasites. It also occurs in transplant rejection and tumor immunity. Example, in the case of an AIDS patient, which had the loss of the CD4 T cells, therefore, there is the reaction of pathogen, or there is the least reaction to the pathogen. So, sorry, which responsible for your TB is merely markedly impaired. What are the characteristics of DHR, which is your delayed hypersensitivity reaction? It is characterized by cell-mediated or delayed type, which is your type 4. It is an interaction of antigen with the primed or mature T cells and represents the damage resulting from inappropriate self-mediated immunity reaction. During mantle test, the cytokines and the gamma interferons activate the macrophage, and therefore it will appear as an indurated or hardened and erythematosus, which is red, which appears 24 to 48 hours after the mantle test. Why it is called delayed? It is delayed because it is used to differentiate between the secondary cell response, which occurs 48 hours to 72 hours, compared to the immediate response, which occurs within 12 minutes upon the reaction of the antigen.
Okay. Agree, disagree? Agree. Very good. Next.
Oh, dali buksoy.